Hello, hi, a very happy good evening. So this is Krishna Ma'am and we're talking about the story of life on earth, and that's biology. So today we are here in lecture 2 of biotechnology and its applications. So we just started off to talk how biotechnology is a part and parcel of our everyday life. So we just came to a point that how biotechnology can be used in our everyday life, and today we're actually going to see how biotechnology is being implemented in the field of agriculture. So biotechnology has been implemented in the field of medicine, in the field of agriculture. So today we'll talk about how it is being implemented in the field of agriculture. Alright, so I hope you guys had a very good weekend, and here we are on a Monday. So I guess once again all your online classes have started, so you're supposed to attend school class from online. So everything is back to the lockdown normal. So I hope things get back to normal and you guys continue your preparation and you guys continue to be safe and healthy. Alright, so chalo, let's talk about how biotechnology is being implemented in agriculture. So before that, I would like to tell you, so please do like, share and subscribe. So please do let your friends know about the session that we are having on YouTube. So yeah, you might ask me, ma'am, why are you teaching only class 12? So after biotech, I will do class 11 also. So I am already done with human physiology. I'm done with genetics and evolution, and here I am with biotech. So next probably I will do plant physiology, and then I'll come back to 12th. All right? Yes. So, and come on quickly, please do like, share and subscribe. So let your friends do know about our session here in YouTube. Yes, please lend us your support as well. Okay, let's continue. So we were talking about how people discovered biotech as a solution. So we were talking about how green revolution came into picture, right? So if you remember how green revolution was one of the solution. So people thought where we are lacking. So we went on to talk about a problem than a solution first. So we started finding out problems so that we can find a solution. So the problem was ever increasing population, but the food supply is less. So this was the problem, and we went on to take up a solution. So we thought about 3 possible solutions, you can say. The first solution was green revolution. So green revolution took into account the chemical pesticides, chemical fertilizers. It improved the existing crop varieties. It used good irrigation practices, etc. Though green revolution succeeded in tripling the amount of food supply, but we also had to consider the harmful effects that it caused to the environment. Alright. So next people went on to do organic farming. So organic farming is where you have zero waste. The waste from one plant or one animal can be used as a manure and other, right? So even green or organic farming took a lot of time and it seemed expensive. So then people went on to genetically modified organism. So when I add or subtract a gene that is genetically modified, so they went on to take those organisms. Okay, so how did the last one help? So that is what we are going to see. So the first type of genetically modified variety was seen in your golden rice. Your golden rice had increased amount of beta carotene, which is pro-vitamin A. So thus people who had one meal could even have still better amount of nourishment. So that is why this was created. Okay. So this is how we were talking about how different methods are being tested and failing. So it is a trial and error process. So finally we landed with biotechnology. So the core research in the field of biology lies in the areas of critical research, as catalyst. So we want a catalyst, something to enhance the speed of the reaction. So that is all what we are looking for. right? So we stopped here. So your genetically modified plants have been used to create tailor-made plants to supply pharmaceuticals. For example, if a plant is giving me an antibiotic, so I would like to increase the supply of the plant, right? So pharmaceuticals increases the production of hydrogen by Brassica napius. So alternative sources to industry fuel, because right now your petrol and diesel are way too expensive, so too expensive to run out for each one of us to have a car. So we are looking for alternatives. So why can't we take an alternative from plants? So plants are easy to grow, that's why we cut them, right? So why can't we look for an alternative? So this is your Brassica napius, right? So we are going to talk about how biotechnology is used in the field of agriculture. It is used in two ways. So what are the two ways? First, to treat cotton plant. So you also know India has a backbone in agriculture, so that is very common. But apart from that, India is also a very strong player in the field of fabric or in the field of textile industry. So spanning from Kashmir in the top to South Tamil Nadu or Kerala in the south, so you have variety of materials, right? So variety of fabric is famous in each place. You have Kashmiri shawls, you have Banaras silk, you have Bengal cotton, you have Bandhani prints, you have Gujarati sarees. So going down, so you have Kanjivaram silk. So throughout we have a lot of fabric that is famous in one country. So we are also a very tough player in the field of textile industry, right? So and India is also famous for its beautiful sarees. So we take pride in all that, right? So when I say so cotton is like a backbone, right? So majority of the things that we use, majority of the clothes that you use comes from cotton. So sometimes it's pure cotton, sometimes it's mixed cotton, sometimes it is poly cotton, sometimes it is cotton blend. So you have various varieties, right? When you shop, the shopkeeper tells you so many names which you have hardly heard of. Yeah. So now if these cotton plants are being eaten by worms or by caterpillars or by small insects, so our yield is reducing, right? So then how do we have a good turnover? So in that sense, so we have to bring biotechnology to give us a solution. So what kind of solution are we looking for? So we are going to employ bacteria. This smartful bacteria is called this Bacillus thuringiensis. So it produces a protein that kills certain insects like lepidopterans, chiropterans and dipterans. So you have beetles and worms. So these are the classes of them, lepidopterans. And then you have chiropterans and diploterans. So these are the different classes, and these are the ones, the worms that eat this cotton plant, right? So this Bacillus thuringiensis produces a particular toxin crystal, which when it enters the gut of these insects causes spores and kills the insect. Thus, it is a very easy easy way that you can kill these organisms. So the gene that encodes this toxin is your Cry gene. So proteins encoded by genes Cry1AC and Cry2AC control bollworms, and Cry1AB controls your corn borer, and this is very important. So what is that I am saying? So we are talking about Bt cotton. So cotton plant is being eaten by a class of worms like lepidopterans, chiropterans, and diploterans, right? Fine. So how do we overcome all this? So we make use of a particular bacterium, so that is called as Bacillus thuringiensis. So during the life cycle of this bacterium, it produces a particular toxic protein in the inactive state. So when these worms inject these bacteria, this inactive toxin gets activated. Due to the alkaline pH present in the gut of the insect. Okay. So now this causes swelling or pores in the gut wall, thus killing them. So my cotton plant is safe. That is why I call them as Bt cotton. So Bt cotton is available as a powder, so I can mix them with water and spray it on the leaves, or I can inject the gene that is coding for this protein. So the gene that codes for this protein is my Cry gene. So from the bacteria, I remove this gene of interest, clone it in a plasmid and put it into the host, that is the cotton plant. Okay, so you have many varieties of Cry gene, Cry1AB, and then I have Cry2AB, Cry1AC. So they all control bollworms. There are certain types of worms that feed on leaves, and then I have Cry1AB. So this controls my corn borer. Okay, done? Fine. So this is about the first application in agriculture, that is your Bt cotton. Alright, so now we move. So here I have, this is your Bt cotton. So the cotton plant that you have seen. So to look into your Bacillus thuringiensis, so what is your Bacillus thuringiensis? It's a typical bacteria which belongs to kingdom Munera, and it's a prokaryote. Right? So now the next problem where agriculture is being tampered. So what is that problem? Is pest, right? So you have plants. So now when I say worms are eating the leaves, worms are pests. You and I agree? Yes. So in that case, we used a bacterium to take over, but worms are actually feeding on the plant, right? So there are actually certain pathogens which can infect the plant. Yes, ma'am, a very, very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. So worms actually feed the plant, so which in turn decreases the yield, whereas sometimes pests or pathogens cause infection to the plant, because of which the yield reduces. In both the ways, you have to save the plant so that our yield increases, right? So the second problem that we identified was pest-resistant plants, and it is hi, after a very long time you are here. So I hope you're doing well. So what are you doing right now? And thank you so much for remembering me and joining me this evening. So please do like, share and subscribe. Yes, Aniraz, I'm doing very well. Thank you. How about you? So I don't see you pretty common here. So you are busy in your own schedule. Well, that's fine. So thank you for joining once again, and please do watch my recorded lectures, and any doubt with biology, so get back to me. Feel free to come and ask me. Yes. Okay, you're repeating NEET. All right. So do you watch my recorded lectures? So which chapter is tough for you? Okay, so now what are we going to do? So you also know a lot of people consume tobacco, and tobacco is a big industry here. Yes. So though you have the dirty picture in front of the cigarette box or on the cigarette box, people still continue smoking, right? So biotechnology is only tough, and I hope I'm doing the right chapter. So I've done biotechnology principles and process just before recently. So you can watch my lectures, and any doubt you can get back to me. All right. Yeah. So pest-resistant plants, so your tobacco is sometimes infected by a group of worms that is called as nematodes. Okay, so one particular type of nematode, so nematode is a common term like bacterium. So under bacteria I have so many varieties. So under nematodes, one particular type is Melanodenine incognita. So it infects the roots of tobacco plants and causes a great reduction in the yield. So one novel strategy how we are going to deal with the tobacco plant and save it from the nematode Melanodenine incognita is by RNA interference. So it is a natural method that we have in our body that is used in silencing of the mRNA. So what does it do? So I hope you understand whatever is here on the slide. So we have tobacco plant, and the roots of the tobacco plant are often infected with a nematode Nematodes are microscopic worm-like structures, but they have a gut arrangement very similar to the humans, okay? So these nematodes infect the roots and causes the reduction in yield of the plant in a way that it causes diseases. So what is the solution that we are going to use? So we are going to use what is called as RNA interference. So this RNA interference is naturally present in our body and it is a type of natural defense mechanism. Okay, so what is going to exactly happen here is, so if you remember the central dogma of life, we have DNA. So information from the DNA is copied onto the RNA. And you should also remember your DNA here is double-stranded, but only one strand's information is copied onto the RNA. So we don't copy the information from both the strands for two reasons. One is that it will complicate the genetic machinery. So number two, it will become double-stranded RNA. But the problem with double-stranded RNA is it will not allow protein synthesis to happen. Right? So if I have double-stranded RNA, which means my protein formation is not happening. So that is why only one strand of DNA gets copied to mRNA. Now we are going to use this technique to kill this nematode in a smart way. Ma'am, how to get your Telegram link? Okay. So you're watching my video. So beneath the video there is a description of my class, like what I'm going to discuss, what is that I'm teaching. So click on the read more option. So when you read more, so you have a lot of information below. So there you have been given an access or a hyperlink to Telegram group. So that is your 𝐒𝐴𝐴RPoint-NEET Telegram channel. So please join the channel first, and once you join that, you can find me as one of the admin. So my name is Krishna Ma'am, okay? So that is how you get in touch with me through Telegram. So you can send me your doubtful questions and I'm ready to get back to you, alright? Yeah. So I am going to form double-stranded RNA so that the important proteins of the nematode is not formed. Alright, so what is that I'm going to do? So I will show you the picture first and then I'll confuse you and then I'll teach you. So I'm going to take the essential genes for the nematodes and I'm going to put it inside the plant. So the genes will be copied to the RNA, but I'll make sure both the strands of the DNA are copied. So if you remember, we call them as sense and antisense strand. So I make sure I get both the strands of DNA converted into RNA. So it will form a double-stranded RNA. But these double-stranded RNA are broken into smaller pieces by a protein called as dicer. So dicer breaks them out into small pieces. So now they are called as small interfering RNAs. Okay, so this keeps circulating in the root of the plant. Now when a nematode comes inside, what happens? The nematode will undergo central dogma of life. The DNA will get converted into mRNA and the mRNA here produces only one strand, right? But we already have the double strand, but this double strand will go here and it will quickly form double strands, right? So your siRNA is double-stranded. This double-stranded is broken into single-stranded by the RISC complex. So everything is single-stranded, so it can go and bind to the essential gene, thus forming the double-stranded RNA, inhibiting protein synthesis. So if you have not understood, I am explaining it here. So we are talking about RNAi. So I stands for interference. So I hope you know the central dogma of life is DNA, which is double-stranded, gets converted into RNA, but only one strand of DNA gets copied onto the RNA and the RNA gets converted into the protein. So protein is in the form of amino acids, right? So when we were discussing this process, we also discussed why both the strands of DNA are not getting copied. If suppose both the strands are copied, then my RNA will also be double-stranded, but this double-stranded RNA will not allow the process of protein synthesis to take place. So here in the process to kill nematodes, I am using the same technique. I take the essential genes of the nematode and put it inside the plant. So when I put it inside the plant, hi friends, yes, good evening. I hope you're doing good. So thank you so much for joining my lecture. So please do like, share and subscribe. So when I put these inside the plant, I make sure both the strands of DNA are transcribed to RNA. Both the strands of nematode DNA are transcribed to RNA. So in a way, I'll form double-stranded RNA. So this double-stranded RNA will be broken into pieces by a group of protein called as dicer. So this breaks them into small pieces like this. So these are now called as siRNAs. Small interfering RNAs. Hi Dharni, yes, good evening. I hope you're doing good. So thank you for joining my lecture today. So please do like, share and subscribe. So how come today Dharni Prince turned up so you are not having school classes? Or did you feel biotech is tough? Ma'am, please tell me how to prepare for NEET. Wow, what a question. So Dharni, are you in 11th or are you in class 12? So I believe you're doing 12th. Ma'am, how to score 360 and 360 in biology? So repeated revision. So read the chapter 4-5 times in NCERT, solve chapter wise questions, solve unit wise question, take up chapter test, improve your speed. So today if you solve 30 questions in 30 minutes, tomorrow you should solve 20 questions in 10 minutes. So that should be your speed. That should be the ratio of your speed. Ma'am, schools are closed till Jan 31st. Okay, fine. So I guess nowadays you guys will turn up for my YouTube class. So you are in class 12. So probably in class 12, biotechnology is an important portion. Genetics is an important portion. Ecology is an important portion. So do the high weightage topics, but at the same time, keep revising 11. So in class 12, suppose if you're studying biotechnology, in class 11, keep revising one chapter like Animal Kingdom. So in a day, you have to do at least 2 chapters parallelly in biology, both class 11 and class 12. Okay? Yes. So, Prince, repeated practice and time management will what help you score. Okay? You don't know how to manage NEET and board. Okay. So, at least make 2 hours time for your NEET. Otherwise, it's going to be tough. So, today probably if you read the theory for NEET, so tomorrow you will solve only questions for NEET exam. So make it alternate. So 3 days in a week you'll study theory, 3 days in a week you'll only solve questions. So that is how you'll have to plan. Okay? Right. So now you have small siRNAs. So now I have the nematode entering the plant. So nematode gains entry into the plant. So once the nematode enters the plant, the nematode will have the central dogma happening. But we already have double-stranded RNAs here. So there's a group of protein called as RISC complex which breaks these siRNAs into single strands. So now the moment the RNA is produced, so the complementary pair will go and bind here and inhibit the protein synthesis. So when the protein synthesis is inhibited, the nematode dies, thus your tobacco plant is saved. Once again, I take the nematode essential genes inside the plant. So when the plant helps the genes to get transcribed, I make sure both the strands of DNA get transcribed. So I get a double-stranded RNA. This double-stranded RNA is divided into smaller pieces by dicer proteins. These dicer proteins now make them into siRNAs. The siRNAs further are cut or renaturated into 2 singular strands by RISC complex. Okay. So once the nematode enters the plant and it undergoes central dogma of life, and when the RNA is produced, my complementary part will go and bind, thus inhibiting protein synthesis. So your RISC complex stands for RNA induced gene silencing. So this is what my risk complex stands for. So this is how biotechnology is being used in the field of agriculture. So first part of the chapter is done. So we will talk about medicine, how biotechnology is used in the field of medicine on Wednesday. Alright. Ma'am, only NCERT, what do you think about notes and modules? So my notes are very similar to your NCERT. For example, NCERT does not mention proteins and risk complex. These are probably extra which you can take note from my class. So you get my PPTs, you get my lecture, so you can make your own notes. And always remember, your notes will help you understand better compared to directly reading from the textbook or taking someone else's note. So stick only to one teacher's way of teaching. So stick to their notes and stick to NCERT. And if at all you find certain extra information from someone else, make a note of it. But questions solve from as many places as you can. So solve from previous years, right? So I take a lot of Menti, make note of those questions. So sometimes I discuss only critical thinking questions, make note of them. So these are ways by which you get exposed to a lot of questions. Alright, so with this I will stop and I'll continue the field of medicine later. So this is how it happens. Alright, so we will stop here, and if you genuinely liked my way of teaching, so please do like, share and subscribe. So thank you so much for joining my lecture today, and I hope I get to see a lot of students because you guys are back to your lockdown normalcy. So please try to come for the live YouTube session so that it can be very interactive. So I'm making very short YouTube classes so that you also have less time and you're also interested to see the video. So it's not tough for me to teach for 1 hour or 1.5 hours, but the point is not that. So whenever the class is short, your mind gets fixed to that class and you have a lot of affinity for the class, right? Assertion and reason, yes. So solve a lot of assertion and reason. So your previous year questions are more than enough because you have a standard answer, right? So please solve previous year question. That is very important. Alright, so please do like, share and subscribe, and I'll get back to you on Wednesday at 4 o'clock. So until then, this is me signing off. So thank you and take care.